Hello, good afternoon, everyone, and welcome back to Carla 2021. Many thanks for staying connected. Remember that you can also follow us on our YouTube channel and on the webpage of the conference, carla2021.org. I hope you have a very nice break, and we now continue with our next invited speaker, Dr. Natasha Sulch. I hope I pronounced this correctly. The title of the talk is HPC in Patient-Centric Fusion of Highly Dimensional Biomedical Data. To introduce our invited speaker, I'll now give the floor to Ulises Moya. Please.
Yes, hi everyone. It's great to be here in this first talk of the HPC plus artificial intelligence conference. And today we have well-known research in supercomputer. She is working in Barcelona Computing Center, and it's great to talk with the Doctora Natasha. Actually, she's in Europe, and she now is late then. And we have one hour to this presentation, including the question. So please, Doctora Natasha, the floor is yours.
Thank you. Thank you very much for a kind introduction, and thanks for inviting me to this conference. I have to admit that it is my first time to talk at a supercomputing conference, even though I'm at Supercomputing Center. My research is applied computer science, design of algorithms for analysis of biological data. And I will today illustrate what we do, kinds of methods, kind of at a high level. Hopefully it will not be too boring and too much of biology for you. And in the end, I will comment on how complex computationally these problems are and why we need to use high performance computing. I hope you see my slides, so if you guys don't see them, then from the organization, somebody should say that now, and I hope they look okay, that you can see the entire screen. Okay, so let me begin. This is an overview of my talk. We view biology and medicine as a complex world of interconnected biological entities that needs to be mined for a new biomedical information that is currently hidden from us in these data. And I will first start with the motivation, and I will give some examples of these algorithms, of these new methods, how to mine these interconnected biomedical data, whether it's a single type of data or various heterogeneous types of data, how we integrate them all. I will briefly mention some of the results and the main techniques. I will also say a few words on our how we apply these methods for the current efforts in the ongoing COVID-19 pandemic. And in the end, I will say a little bit more about the computational complexity of some of these methods that we develop and how much time we take on our supercomputer to solve them. And in the end, I will conclude. So let's begin. So technological advances have yielded an astounding harvest of various molecular and clinical data. I'm sure you've all heard of the human genome, and then there is the epigenome, which is how our genome gets modified based on our lifestyles and that we actually pass on to future generations. Then there is transcriptomic data, meaning that because each cell has a full copy of our genome, but we have skin cells and brain cells and heart cells, etc. And it's only which genes are turned on and off that differentiates these cells and tissues. So this is so-called transcriptome data. Then there is a proteome data, meaning the set of all proteins in the cell or in the human body, which is very many of them, because genome is only the blueprint of the cell. It makes instructions to make proteins, and then it's the proteins that do everything in our cells, actually. Then you all know about the metabolomes, right? When you go see a doctor, they measure things from your urine or your blood. These are metabolites, right? That you produce. Then there are all sorts of exposures through our life, all sorts of toxins, whether we smoke or not, consume alcohol, etc. Then there is metagenomes, which means various kinds of microorganisms that live on our skin. We are covered with them and live in our gut and help us metabolize food, produce vitamins, etc. All of these are huge, huge data sets. And this data growth about a cell of a human body has now made us hit the wall of biocomplexity. This is very complex data because they're not just isolated doing something by themselves, but they're very intricately and precisely integrated into networks of interactions between these various molecules, these genes, proteins, metabolites, etc., within our bodies, and also interact with the environment. And all of these different data types, basically, it's like putting different pair of glasses to look at the same thing, at the human cell and the human cell. It's just different layers through us, actually. So this is why all of these different measurements, whether it's genes, proteins, metabolites, and their interactions, they complement each other, and they need to be modeled and mined together to get a full picture of what's going on. And this is what we do in our lab. So this is a relatively new research line here to integrate, to fuse all of this within a mathematically principled framework where all the data fit together. And then basically to analyze them, you need everything from computing and algorithms, including machine learning, mathematical nonlinear optimization, network science, or psychology, etc. And because the data are so large and complex, you need to do that using high performance computing. And we do all this modeling and computation on all of these data that machines produce, robots produce for us, to enable so-called precision medicine or personalized medicine, so that we wouldn't just get, you have this disease and every patient of the same disease gets the same prescription, but which you might not react at all. Actually, we want to precisely treat every individual based on their genetics and exposures, so to do more precision, both diagnosis and treatments. And as I said, everything is very computationally complex. We need high performance computing, and I'll tell you a little bit more about that. And I have to acknowledge a big funding that I received to do this ERC Consolidator grant. This is a single PI grant. These are the most prestigious in Europe. It's 2 million euros over 5 or 6 years. So these results come from there. And previously I had an ERC starting grant just before my ERC consolidated. That was 1.6 million euros. And that actually enabled me to move my lab from America, from the University of California, actually to London first, and now to Barcelona, and do this research in Europe. And recently, last year, I also got an ERC proof of concept grant to try to commercialize basically the results of this research. Okay, now what are the computational challenges? So basically, you all know that we've had the human genome since year 2000. This is when the first version was published, both by a public consortium and also private. Celera company did this effort. And even though you could view the human genome as a linear sequence of letters A, C, G, and T, and even though to align or compare these linear strings is computationally easy, meaning you can solve this problem of aligning or comparing in the time polynomial in the size of the input of your data, we still don't have a very good understanding of our genome, its function, what it does, what gets broken in disease, how to fix it. And in addition to that, dealing with large network data, these networks of which gene influences expression of another gene, or which protein binds with another protein to do a particular function. This is much more complicated. These problems fall into the category of computationally hard, computationally intractable problems. You all know NP hard, NP complete, etc. These hierarchy of computational complexity. And even a simple thing that you might think is simple, such as aligning or comparing two networks, this is computationally intractable because of the underlying subgraph isomorphism problem. So the only way to solve this on these large data, we cannot do it. And the only way is to make heuristics or approximate ways, approximate algorithms to approximately say whether these networks are similar or not. So basically, this is what we do in the lab, and this is the reason why we do that. We make these approximate methods, heuristic algorithms, but they have to be sophisticately tuned to extract new knowledge from particular data, because every heuristic, every machine learning method, they're all heuristic, they're all going to fail on some data. This is what computationally intractable means. So you can only solve them well, approximately, on a particular data. This is why you have to really be tied to the data, and everything genetic will fail, as you all know. And then we have these various layers of networks measuring different things in our cells, whether it's genes that are turned on or off at the same time, or whether these proteins that bind together to a particular function, or whether it's different metabolites, their interactions, or whether you have the network of drugs and you can link them by their side effects if they cause the same side effect, or if they're chemically similar, etc. There are many ontologies, disease ontology, for instance. If you have a particular disease, whether that makes you susceptible to another disease, for example, diabetes, you're much more susceptible to certain kinds of cardiovascular diseases, but certain diseases protect you from other kinds of diseases, right? So this is so-called disease ontology or gene ontology. This is our model, how we think biological function of genes is organized. So you have all of these various networks. So we want to extract as much information out of each of them as possible, which is already very hard computationally, but also we want to see what they collectively tell us. And not only what they collectively tell us. I'm sorry, the animations are not coming through correctly, at least through this Zoom that I see. Hopefully for you it's better. So not only what they tell us collectively, yes, this is an even harder problem, but also how to put that in a patient-centric way to solve the tasks, to basically make our medicine precise. So this is precision medicine. Okay, so this is the motivation. This is why we do what we do. And now some examples of the new methods, how to mine these interconnected. So the first kind of methods I will show are from my earlier work, because I use them later. This is why I'm showing them. I'm using them later in the second part of the talk. So how to mine one type of a network, one type of molecular data to learn about function of various genes or proteins, their involvement in disease, in various diseases, whether it's cancer, leukemia, cardiovascular, rare diseases, or more recently, COVID. So let's start with that now. One of the main workhorses of the cell are proteins. So proteins are three-dimensional molecules that usually don't do anything by themselves, but by binding with other proteins, so physical binding by physical forces in proximity, if they have complementary so-called binding domain, one looks like a ball, the other one looks like, like this, and they click together. So when they bind, they change their 3D shape, and that change of the shape enables them to do a particular function. For instance, they can maybe act as scissors to cut the proteins that are damaged or at the end of their lifespan, to reuse the little bits and pieces of those proteins to make new proteins, for instance. So this is what a protein is. So they do pretty much everything in our bodies. And to abstract from all of this complexity of their 3D shape, etc., we model them as nodes in these so-called protein-protein interaction networks. And if they can physically bind, we put an edge in between. And then presented with one network, protein interaction network, let's say of a healthy cell and a diseased cell, we want to compare them. And I just told you in the introduction that this is computationally intractable. So what do? We measure simple things. These are these heuristics. So for example, we can measure how many nodes and edges these networks have. And if they're similar, we say, these networks are similar. Or the number of edges that a node touches is so-called degree of a node. And then you have the distribution of these degrees over all nodes. And this is the degree distribution of a network. And you compare degree distributions of two networks and to roughly say whether the network structure is similar or not. But all of these, as I told you, all of these heuristics fail on certain examples. So this is an example of two networks, G and H, of exactly the same size, the same number of nodes and edges in each one of them, exactly the same degree distribution, because each node in each network has degree 2. It's linked to 2 other nodes. And you can just eyeball them and see how very different they are. One is connected, and so the signal will pass through eventually, and the other one is disconnected. The signal cannot pass through, for instance. Basically, to constrain the topologies, the structure of the networks as much as possible, in 2004, I invented graphlets as Legos of networks. And basically, I'm asking, by the way, these little substructures, so I enumerate all of them. This is the only one with 2 nodes. There are 2 of them with 3 nodes, a path and a triangle, etc. So these are all of them with 4 nodes. I'm asking whether, by the way they are put together in a Lego fashion in these networks, whether we can figure out what do we have made of these Legos, whether it's a helicopter, whether it's a pumpkin, right? And this was a subject of my ERC starting grant some time ago. And you can even see, like, basically, if you want to enumerate all of these in large networks, let's say if a network has n nodes and you want to count all up to 4 node graphlets, your complexity is n to the 4th. For large data, this is prohibited. And I'll show you in the end examples where even when we have linear algorithms, we have to use HPC for you're just loading and doing simple things on this data. And if you want, for example, 5 node graphlets, then it's complexity n to the 5th, etc. And then, but to ease this, not just to count, to enumerate these, then we notice symmetries within these graphlets. So for instance, n nodes of a 3-node path look the same, but the middle node looks different. And these are so-called automorphism orbits, etc. Let's not go into the mathematics of it. But when we enumerate all of them for up to 4 node graphlets, we get 14 orbits. And what do we do with them? So let's say for this node A in the network, we're going to make this feature vector of how many of each of these graphlets it touches and at which location. So for instance, this node A touches 4 edges, 1, 2, 3, 4. This is why this coordinate of this feature vector is 4. Or for instance, it touches 3 node paths at an end. This is the first one, this is the second one, this is the third one, etc. It touches 1 square. This is why this coordinate corresponding to the square is 1. It touches 1 triangle. This is why this coordinate corresponding to the triangle is 1, etc. And then we do this for all nodes, and we have these matrices, so basically the rows are these feature vectors corresponding to the structure, the wiring pattern around each node in the network. But then if you go along the columns, let's say this first column is your degree distribution that I just mentioned earlier on. And this is orbit 1 distribution, orbit 2 distribution. So you can tell about the wiring pattern of the entire network. And then you manipulate this matrix in various ways. And then we do many things by playing with this matrix. For instance, the network alignment problem that I told you about. We want to take, let's say, diseased network, let's say in cancer and in healthy, and to compare them, computationally intractable due to subgraph isomorphism. And then, we use the graphlets to do that, to, seed, anchor somewhere, extend, etc. And this is, and basically we've shown, we have a series of so-called GRAL algorithms for graph aligner, where we have shown that almost the entire protein interaction network of yeast exists as an exact copy in human, which is interesting that species so diverse as a yeast, baker's yeast, and human share that much wiring of the wiring structure. We've also early on shown that the similarity in the wiring patterns around proteins in protein interaction networks, similarity in biological function, similarity in doing something in cancer, different kinds of cancer, etc. But let's not dwell onto the biology much. And basically, this wiring pattern is linked to biological function, even in diverse species. For instance, on the cell membrane, there exist proteins that sense the environment or take the nutrients in or a signal into the cell. And then these proteins on the cell membrane, in diverse, from yeast to human, they look like these hubby ones, as you see here, red on this membrane. They take stuff in. But for instance, these proteins that are so-called transcription factors that enable the transcription, the expression of genes, they look more like cliques, like fully connected graphs, and this carries across life on Earth. And also, as I already mentioned, is that cancer genes have very particular wiring patterns around them. So if you could detect some genes for which you don't know, that it's not known that they have anything to do with cancer proteins that you don't know, then by wiring pattern, you can say, okay, this might have something to do with cancer. And this is very important to discover such proteins, because basically it's proteins that drugs bind to. So basically then you want to stop cancer at a certain, or Parkinson's disease or whatever, right, at a certain path through it, so to disable this disease. This is our more recent work from a year ago, that we published in ISMB, Intelligent Systems for Molecular Biology. This is our top conference with acceptance rates of about 15%. We analyze the data of leukemia, of the basically leukemia cell and the healthy cell of origin. And this data is another kind of genome data. This is again new data because the genome is very long, really, really long string of letters, but it doesn't exist as a string. It's packed. It would be like 1.5 meters long, but it's all packed within the nucleus, but in a very particular way, very modular way. And this is new chromatin. This is called chromatin. This packing nucleus. And then we analyze the structure of this chromatin in the nucleus of every cell. And in the healthy cell, you see the modularity. You see, the structure. But in leukemia, this is completely disrupted. So there's no structure anymore. But okay, biologists have known that cancers of all sorts rearrange how your DNA, how your chromatin is packed. But what we have found that is interesting by using our graphlets, these are those orbits and orbit counts in the driver genes of leukemia, meaning these are the genes that when you get a mutation of them, they drive the progression of the disease. This is at least what's believed, right? So we see that there is differences in the structure of the packing around these particular genes even before cancer hits. So basically by looking at the chromatin or the healthy one, you could predict where a particular cancer would hit and try with medication or nutrients or lifestyle to protect these particular regions. Well, this is for now our hope. We cannot do that yet, but we are making the first steps. So anyways, everything you've heard about graphlets, we've generalized to directed networks as well. We can track the dynamics of how nodes change to different networks, not only in biology, but in economics as well, in world trade, etc., or how the networks change through time. And even early on in 2004, and then I confirmed in 2007 in my single author paper, again in our premier conference, European Conference of Computational Biology, with 18% acceptance rates, I've shown that these protein interaction networks have actually geometric structure. Of course, it's not random, but it's not even scale free, like everybody was talking 20 years ago about, but it's actually geometric. And we'll see now that everybody's talking about geometricity of the data, embeddings in particular spaces, topological data analysis, etc. But actually in 2004, this was the first mention that these data are geometric. Also, last year in ECCB, we generalized graphlets to work with weights, with probabilities on the edges. And we have shown that by incorporating these probabilities, we can better identify biological condition-specific functions in biology. We have then generalized Laplacians to graphlet Laplacians, generalized spectral embedding and clustering to work with our graphlet Laplacians, and shown that we can use them to detect different and complementary biological annotations, meaning biological functions. And others have used graphlets, so they received really high citation counts. They've been used for image categories recognition, scene classification, photo cropping, then also in social networks for personality and affective states recognition in large organizations to maximize productivity, etc. Now, I'll just briefly mention how it's not just pairwise entities that interact in the cells, but we have multi-scale hierarchical organization and how we deal with that. For instance, as I told you, proteins bind together to do something, and not only two, but many of them. And this is called protein complex. So let's say when we have this protein complex of 3 proteins, we model it as these 3 nodes and edges in between. But also, if we have 3 protein complexes of 2 proteins each, we model it in the same way. And basically, by modeling, you lose information. And a better way to model them is by hypergraphs, clearly, because you have nodes, and then any subset of nodes can be an edge. And here, clearly, this 3 protein complex is modeled by this set of 3 nodes, and then 2 protein complexes by these 3 sets of two nodes each. And hypergraphs, until our research, was not used much in systems biology. Centrality and clustering was generalized from graphs to hypergraphs by Ernesto Estrada. Also, degree distribution and some general modeling was considered. But basically, we took this protein-protein interaction at first that I told you about, took pairwise interactions, but also all of the protein complexes as hyper edges, and also pathways. Pathways are these things throughout the cell, how the cell reacts to signal, how it senses the environment. Let's say, it detects sugar, it says, okay, there's sugar, I should take in sugar, sends the signal through the cell to turn on in the nucleus certain genes, to make enzymes so that this sugar can be utilized in the cell. So these are so-called pathways in the cell. So we model all these by PIH, we call it PIH, protein interaction hypergraph model, for this multi-scale organization of biological data. And then everything you've seen for graphlets, we do with hypergraphlets, and orbits with hyperorbits. And we get these very long feature vectors. Even for just 4-node hypergraphlets, our feature vectors, counting all of these orbits in them, are more than 6,000 long. These vectors are 6,300 something long, coordinates long. And then we again cluster these, we compute all of these feature vectors, we cluster nodes, i.e. proteins based on them. And we find out interesting things, that certain biological functions dealing with, so proteins dealing with learning, memory, also amyloid fibril formation, neurodegenerative diseases, synapse organization, and other neuronal functions, they all look like these. They're characterized by these orbits. We don't know why, but for now we are noticing patterns. Hopefully one day we will also know why. And of course, with our model of multi-scale organization of these data, we extract much more biological information than by a simple pairwise interacting models. And then we've used hypergraphs to make kernels for classification of biological networks with collaborators from the US. Also, a similar notion, but different, is abstract simplicial complexes from algebraic topology. We generalize graphlets to simplets in these terms, and did the same as we did for hypergraphlets, and we get more biological function out of it. And why we want to learn biological function, because about half of the proteins or genes in our body, we know they are there, but we have no idea what they do. Even for baker's yeast that we use in bread every day, we know mostly what they do. For that, recently we figured out, but for human, about half, we don't know what they do. And we need to know what they do if we want to understand our bodies and cure our bodies. Okay, so let's stop talking about these examples of how we mine one type of data, proteins, let's say, and their interactions. But now we have heterogeneous data. So I will illustrate on a couple of examples. I will talk about iCell, and we published this in Nature Communications in 2019. As I told you, different kinds of molecular data complement each other. This is why we want to collectively now analyze them. So we've taken gene co-expression data, so whether genes are turned on at the same time in the same tissue, but this is one type of a network. Then protein-protein interactions, another type of network that we took, and so-called genetic interactions. These are epistatic interactions, meaning we did double mutants. When you mutate one gene, nothing happens with the cell. When you mutate the other, nothing happens. But when you mutate both, something unexpected happens. The cell gets sick or dies, or maybe proliferates uncontrollably, etc. These are so-called genetic interactions. So there's a whole network of these genetic interactions. Right? So we did these three kinds of data for cancer, in cancer tissue, and in healthy. Then we integrated them, and I'll tell you how in these integrated cells that I call I cells. This is a new model of biology based on these data. And then we compare them using graphlets, because this is the best method we know how to compare networks. So we compare them to find new cancer genes, and we got biological validations that these are truly new cancer genes. So how did we do this? And why we needed to invent a new method? And what I mean by the claim that different data types complement each other is the following. So we took these three different kinds of data: protein interactions, co-expression, and genetic interactions. These are all on genes. They're all networks on genes. And we see that they have around 3,000 genes in the overlap. But look, they have only one shared interaction in the overlap. And there is nothing wrong with this. It's just we measure different things by different technologies. It's not the same whether they're expressed at the same time, or whether they bind, or whether they epistatically interact, etc. And this is why, because of this one shared interaction, most of the previous methods fail. They either diverge or stop before finding clusters, or don't produce any clusters, or produce very many very small clusters with which you cannot do anything, etc. And this is why we put these kinds of data. Let's not go into this biology. This is not a conference for that. So we took these different kinds of data for 4 most common cancers in human: breast, prostate, lung, and colorectal. And these are the control tissue. So these are healthy, proxy for healthy. And we made these networks. And you can see both for control and cancer, these are large networks. They will have around 10,000 nodes and around 100,000 edges. So these are fairly large networks. So let's say we have these 3 types of networks on the same kind of entities. Whether it's gene or a protein, we take them interchangeably because genes make proteins. So this is why we kind of say they're the same, even though really they're not, but this is a good enough approximation. So we make these networks as matrices, normal way to represent any graph, any network. Gene A, if they interact with the gene B, then there is a value of that interaction or expression or something. So we have matrices corresponding to these 3 types of data on the same kind of entity on genes. And then we approximately decompose them into 3 lower dimensional matrix factors by minimizing this objective function, AI minus GI times S, G times SI times G transpose, sum of that. This is Frobenius norm. And then we get these Gs. So this is low dimensional, but this is all of the genes, but this is lower dimensional to gather the information out of it. Now we multiply G times G transpose to get another gene by gene matrix that we call I cell, or an integrated cell. And then we find clusters in them, and you see by the same color, I colored the same function, the same biological function. So we can see here that in these I cells, we have more enrichment in these clusters of the same biological function than in any of the constituent data type alone. And then that this I cell is non-random. We don't know which model it belongs. So we didn't study it for now. Hopefully we will. And that this is the percentage of these clusters with enriched genes, genes enriched in a particular biological function. And you see when you add all data. So this is if you only use protein interaction data to do this decomposition, or when you use protein interactions and co-expressions, etc. But with all of them, you get the highest enrichment in the same genes of the same function within these clusters that we obtain here. So there is biological signal in it. Then we created these tissue-specific networks for cancers and controls. And only the genes, not the genes, sometimes cancer silences particular genes, turns them off, or expresses genes that are not supposed to be expressed. But only these genes that are expressed in both cancer and control, we found that they are enriched in these driver genes that, if you get random mutation, usually nothing happens, but when this mutation, this driver gene happens, this is usually when you get a disease, right? So these always expressed genes are enriched in these drivers. And then we found out that when we compare between cancer and control, these eye cells, this new eye cells, new model of molecular network, and also the constituent networks of data, the real data, protein interaction, genetic interaction, co-expression, only in eye cells, we measured what was rewired, control versus cancer, and how much there was a rewiring. Basically, only in eye cells, we found that rewiring is indicative of oncogenicity. So we took basically this as a signal. We found 20 of the most rewired genes in cancer eye cells for these 4 cancers. So 4 cancers times 20, there will be 80 genes, but some of them repeated over different cancers. So we had only 63 unique genes. So these rewired in eye cells. And then we basically validated almost all of them either through literature curation, that there was some paper mentioning them in that they might have something to do with cancer, or by our own knockdown experiments where those genes, basically we silence them so that they are not expressed, and then or we overexpress them. And then when we did that in these cells, we observed whether the cell dies or what does it do, right? And then we observed either significantly reduce of these cells with these genes knocked down in cancer, or increase. So they do something. We biologically validated that 17 out of these 63, that we validated a large number of them by these knockdown experiments for cell viability, and also by retrospective analysis of patient data. So basically, patients in mutations in these genes have different survival. Some live longer, some live shorter, etc. And these 17, only 17 of 63 are so-called differentially expressed in cancer. So they're expressed, let's say at a certain level of in a normal tissue, but more or less in cancer. This is so-called differential expression. So you could not have even identified them with these differential expressions. You cannot identify them in protein interactions or genetic interactions or by co-expression or differential expression. So basically only in this fused model you find these new genes. And basically we repeated this on 16 more cancers, kind of pan cancer, everything that we found in TCGA, 20 cancers in total, and we did the same. And for instance, this top one, this gene, it was not known to have anything to do with cancer. And we found out that in 8 different cancer types, patients with this gene mutated has different survival. So basically, this is what our integrated thing does. But then we will see that it's computationally complex, of course, to decompose these matrices. How do you find this low dimensional K that's optimal, etc., etc. All right, so I would conclude here that basically we need this data fusion to find important things in important diseases such as cancer. And then we did this in other diseases, including rare diseases. This rare form of thrombophilia. This is rare diseases. Basically, you have very few families with this disease, but it's nasty. So here, for instance, we have 2 brothers, one diseased, one healthy. The healthy brother has a daughter that's diseased. That's one family, and the second family is 2 sisters. So if you wanted just to learn something from genomes that is statistically significant, you cannot. You have very few samples. So this is, but then when you overlay with networks and do this data fusion, again, this minimization, then you boost the signal. And we found out that, a cluster of genes that are mutated in all disease women, for instance, and this one that is mutated in the healthy brother only. So probably these mutations protect this guy, so he's healthy. So this is, we submitted this work with collaborators from the Institute of Genetics in Belgrade, because this is where this rare mutation happens, or the Belgrade mutation. Let me just show you. So this is what I've shown you, this matrix factorization. This is an AI method. You minimize an objective function, you have some rules, how you do that. The reason why we like it, it's because it's an explainable AI. It provides mechanistic explanations. And here, so far, what I've shown you is on genes. We had 3 different kinds of networks on genes, whether it's protein interaction, genetic interaction, etc. But then, patients would have certain mutations. So there will be this matrix of patients, and which patient has a mutation in which gene. This is a gene in this, this is a patient. So this would be this B13 from patients to genes. There are somatic mutations, for instance. And then patients could take particular drugs for the disease, or have taken. And our question is this good, or should we give them different drugs? This is so-called drug repurposing. And then also drugs bind to certain gene products, to certain proteins. So basically, protein is a 3-dimensional molecule. If the gene is mutated, it makes faulty protein, but then the drug binds to it to make it in the original shape, and to restore its original function, right? Or actually, more often, it inhibits the function of that protein, which basically then means it's a poison, right? So it inhibits the function of a protein, so drugs in effect are little poisons. But let's get out of this philosophy. So you have drugs, and where do they bind? To which proteins do they bind? So there's this matrix D12 across, right? And then you can decompose these matrices across again into three factors, where you, through decompositions, keep some of these factors the same, like kept G the same for those decompositions. Here is G1 corresponding to genes, it's the same color, red and red. G3 corresponding to patients, this is why it's green, and G2 corresponding to drugs. And this S12, S13, S23, these are low dimensional middle factors that are there for technical reasons. So we minimize this objective function, and then basically this sharing of the factors enables the fusion of these data. And then when we want to repurpose this drug to this patient, let's say this dashed line, we can say, okay, we want to repurpose this because this particular patient has these 2 mutated genes that make faulty proteins that this drug binds to. This is mechanistic explanation. This is why we develop these methods, even though they are computationally complex, but then we use sparsity of the data, parallel matrix manipulations to handle the complexity. Okay, and then basically this is just an example that we published as early as 5 years ago in the Pacific Symposium of Biocomputing, when we took from the Cancer Genome Atlas, the TCGA, 300-something patients, all of the same ovarian cancer type, serous ovarian cancer patients, and then their somatic mutations, the mutations of these patients in these particular genes. This is this matrix R12 for TCGA. These are real data. And then on the genes, we found protein interaction at first, genetic interaction, and here metabolic interactions we took also between enzymes. And then from drug bank, we found drug target interactions, and then we basically decompose these matrices across R12 and R13 into three factors here. And basically from this minimization function, after it converges, right, we read off 3 tasks of precision medicine at the same time, at the same computational cost. This is patient stratification. It comes from this cluster corresponding to patients. This is matrix corresponding to patients. Then cancer gene prediction comes from the cluster corresponding to genes. And then these reconstructed entries that come from this fusion, these are predictions for which drugs to repurpose to these patients. And then again, an illustration that if we include all of the data in the fusion, we get the highest area under the the RO curve and precision recall curve and the smallest standard deviations. We apply this to the new data of the pandemic. As early as April last year, we got already biological data on the interactions of how the viral proteins to which human proteins they bind. So these are so-called viral host, human host interactions. So we had these viral host, these are SARS-CoV-2, this is the viral proteins, and to which human proteins they bind. On the other side, we took all of the approved drugs and their bindings, drug target interactions to the human genes. So why we did this? We did this because antivirals are difficult to make and it's questionable how well they work. We've seen a few days ago, Merck announced that they have a new antiviral for COVID that works very well, which I really was very happy to hear, but I was a little bit sad to hear that they stopped the clinical trials on it. Let's say I didn't read it very carefully. I was very busy these days, but I intend to. So anyways, we want to move away from antivirals because viruses have very few proteins. They have like 30 something, let's say, proteins, but humans have many, many proteins. So the idea is to go into the human molecular interaction network and interfere there with the drug to prevent the viral processes. So this is, basically we took the data, we did the same minimization, fusion of all these data that I illustrated, I will not repeat. And what did we find? We found what is interesting here is these drug target interactions, the new ones that we predicted should be given, these drugs that are already approved, but for different kinds of patients, but we predicted they should be given to COVID patients as well to improve their health. Where are they? They were not basically the sweet spot where you should target is not these human proteins that directly interact with the virus, nor these proteins that are directly here targeted by the drug, but the common neighbors. So basically, it's the network neighbors of viral targets and a differentially expressed gene. One kind of data, I forgot to mention this. So one type of data is the SARS-CoV proteins binding to human, but another one is in the lung of the human, what gets differentially expressed in these patients, in COVID patients. And usually you want to target with a drug either the viral, the direct interactors with viral proteins or these differentially expressed ones. But we found out, no, our drugs don't go to those, but they go to common neighbors of these two. And then we found out that these common neighbors have very different network properties, much larger degree in the network, also to centralities, etc., clustering coefficient, and also graphs, basically all but 4 orbits are very different in these common neighbors. And if you want, you can read more in this paper that just appeared recently in scientific reports. And the bottom line here, when I would stop talking about this biology, why is it important? What are we contributing? I would just like to say that within this framework, you can put anything you want, any data that you want, any DNA elements, treatments, adverse effects, I don't know, all sorts of things that can happen because these epigenetic marks, clinical profile similarities between patients, etc., etc. But each time you do this, I forgot to mention this. Basically, this minimization corresponds to different NP-hard continuous optimization problem. You have to propose objective function. You have to find this optimization solver, proof convergence, correct. And optimization is slow, so we have to use high-performance computing. And just some illustrations of how complex these these things are. So this is, again, patient-centric fusion for highly dimensional data, but this is, for instance, for Parkinson's disease. We have collaborators in Luxembourg, Alexander Skupin's lab, that basically measure these neurons from pluripotent stem cells, differentiate them into the neuronal cells, and they measure what happens, various kinds of measurements, these expressions, etc., interactions, at different days, day 5, day 6, day 15, day 21, etc. And so we want to fuse, and this is at the single cell level. This is not bulk data. So for each of these cells that differentiate, differentiate at these time points, they measure. So this is a lot of different cells, and we want to fuse this single cell expression transcriptomic data for Parkinson's disease with molecular interaction data, and we need to perform NMTF, non-negative matrix repastorization, around 1,500 times. We use multiplicative update rules. We have different computational scenario corresponding to these cell conditions where they are, both in control and in the disease, in the sense that these are the cells with mutations that we know are in Parkinson's. So the complexity is this, if you can read this. So basically, N is the number of rows and columns in any of these adjacency matrices that we're fusing. M is the number of adjacency matrices. K is the number of clusters that we want to find of these genes. We want to find we want to find the disease mechanisms, why Parkinson's progresses, where, at which day it progresses, and how to hit that with which drug to stop it, right? So this is quite complex, I would say. P is the number of possible combinations of the input data, etc., etc. The size of the input data we have right now is 0.5 GB in the text files. So currently on HPC resources, this takes us around 750 hours of CPU time across 40 processors for each of the 64 computational scenarios. So 64 times this. Okay, but now this is an even worse example, another example. This is SNP-SNP interaction data. These are different mutations, and whether they occur in the same patient. This is from GWAS studies, from UK Biobank for diabetic patients. We want to find, so basically, you know, we know these SNP-SNP interactions, but just in pairs, but nobody's asking, maybe it's not in pairs, maybe it's in triplets, or maybe, you know, there's some communities of these. But how to do this when we have a 300 GB text file containing 20 billion entries? You know, so we need HPC to load and analyze them. And basically, due to the size of this data, we have to use linear algorithms. So basically just heuristic, nothing other than linear can work. And another example of these HPC problems is, and the last three examples is ongoing work of my students right now. So I don't want to give much detail just to illustrate the complexity, because it's not finished work. So it would not be fair to them to talk about that. But for instance, there's another problem when we want to embed biological sequences. We want to go beyond just aligning sequences because that's limited. We've learned a lot with it, but we want to learn more. So we borrowed from NLP, natural language processing. We identify different k-mers, different length K along these sequences. We have a sliding window. We go like in NLP through it. And then, for instance, you know, if the k-mer length is K, so 10, where we want 10, you know, the length of this genome ACGD of size 10, and then to see if they co-occur in a certain window size, then basically, and we want to make this positive point-wise mutual information matrix, PPMI matrix, and if L is the length of the genome, W is length of the sign, is a sliding window, K the length of the k-mers where we are finding these co-occurrences, this PPMI calculation of this matrix, this is its complexity. L times W times 4 to the K, exponential. And the human genome has 3.2 gigabase pairs, right? So for k-mer of size 10, here is the matrix size. We take 200 hours on 2,400 CPUs so far.
Okay, we have 5 minutes more.
Okay, this is basically the last slide.
Okay, thank you.
Sorry, this doesn't show me any time, this Zoom. And then we want to also to do embedding, and the complexities and cubed for the embedding. And the input data PPMI matrix compressed is 600 gigabytes. So basically it would be 4,500 gigabytes uncompressed. And we don't even know how much resources we need for that. So to conclude, want to mine all data to learn more about disease, how to cure disease, how to even extend the length of human life. We need paradigm shifts for that, both computational and methodological. In terms of computational, we want to analyze all data within the same framework, new bottom-up data-driven approaches, biological concepts, etc. We've seen one first prototype iCell that I talked about. Others are doing that. There was a very interesting paper earlier this year, midsummer, about the multiple paths to multiple life. Physicists from Santa Fe, basically, they say one layer is materials, biological molded materials and biophysical laws. Another one are constraints that come from architecture, dimension, topology, etc. Another one is measures of information, free energy. So people are thinking in these ways. What is biology really? And then we need methodological formalisms, even when they exist, to model multi-scale organization or dynamics, etc. We don't have algorithms to extract this information. We've seen some examples of algorithms such as hypergraphlets or simplets, etc. And then there are so many tricks we can do to make these algorithms efficient. Hopefully algorithms to analyze this data, but let's not talk about that. No matter what we do, computational issues will remain with us unless somebody proves P equals NP. And this is why we have to train people and embedded data scientists that speak all languages, medicine, biology, in this case, computing, AI, HPC, etc. To that end, I just published a book a couple of years ago, a textbook for all of these domains on these things for masters and PhD students and new scientists. So, and to take all of this out of the academic setting, I believe this is important. And we have this grant. We have set up a small startup, still doing nothing. We're looking for investment. Let's see if we can manage. We don't have any credibility here. We're just researchers. And I need to thank these ERC grants that support this research. Also, to thank my group members. I have 13 members right now in the lab. Unfortunately, there's no new group picture because we've been working from home for a year and a half now, mostly. I thank my past members, many collaborators, and also learned societies such as Academia Europaea, British Computer Science Society, and the Royal Serbian Academy of Scientists and Artists for admitting me as their members. And thank you very much. I hope this was not too boring for you and I can take some comments and questions.
Thank you very much. It's a great presentation. And yes, we have a couple of questions here. One is this: do you use Jupyter Notebook to present the output graphlets, or what do you use to present the graphic output?
Yeah, we do actually. We have a research engineer in the lab, Carlos Garcia. This is the guy here. And actually right now he's building a platform in which he would unite all of these into one platform. We would make that available because this is all research software. We make them all available as we, with each paper, but now it will be as part of it. Yeah, we do. But I would refer you to Carlos.
Thank you. And yes, we have another one here. And this is, could you apply these techniques that measure and detect, I don't know this word, to other illness such as long COVID, or in addition to cancer?
Do we detect other illnesses, you mean, for other illnesses? Oh, yeah. COVID, you've seen an example, various kinds of cancers, ovarian, colorectal, leukemia. We did cardiovascular disease, as you saw, a rare form of thrombophilia. Now we are continuing collaboration with the Genetic Institute, actually the Institute for Molecular Genetics and Genetic Engineering in Belgrade, basically to find the link of, they believe there is a link between thrombophilia and the colon cancer, that same things are involved. Oh yeah, you can apply to many different diseases, but not only in biology. We apply them, for instance, to economic networks, to the networks of trade, and we have interesting results there. But this is not our main thing, This is when we have a willing collaborator in economics.
Thank you. We have another one. What kind of GPU do you use for your calculation?
We have, yes. GPUs. We have not used GPUs so far. We just use normal, cluster computing. Not the problem is memory, and we are struggling even on with very large memory. We have to cut these matrices and then recombine them, compute and then recombine. So this was the main reason, but probably there is a way to utilize them, but no time to do everything, even with 13 people.
Okay, thank you. I have one question, one last question, I think, about this. I don't know if you have used these new techniques called geometric deep learning and all these things, all these tools for graph representation, non-Euclidean representation. Do you use these kind of tools?
No, we have not used geometric deep learning yet. And the reasons are quite technical. I would not go into the details yet. We basically believe that with what we have is we can achieve more, because with these graphlets, we go much deeper into the structure around. And also with, drawing that with these factorizations. Intentionally, we chose factorizations. I mean, some of these matrix factorization approaches are equivalent to deep learning. You can show that, I mean, there is theoretical proofs for that. But basically, the way we do it, that enables, within the same framework, very good data fusion that sometimes it's not possible to do with these other approaches. I mean, we are aware of them, but we choose to do particular things.
Okay, thank you. Thank you. This is a great talk, great questions. And we want to thank you again for the Carla organization. It's a pleasure to talk with you and hear your ideas and your work. So thank you.
Thank you.
I don't know. And see you in the next conference. Yeah.
Sure. Hopefully in person. Bye-bye.
Great presentation. Thank you for your attention. We invite you to stay with us and to continue with our next sponsor talk by Red Hat. The next moderator is Efrain Sanchez. He's a solution specialist for public sector in Red Hat, and he will introduce our next speaker. And now I give the microphone to Juan Carlos Rosas. Juan Carlos Rosas.
Thank you very much. Thank you very much for this presentation. I have the honor to press the Red Hat talk, and Ronaldo Cedillo, and solutions architect, and Efrain Sanchez with us, especially solutions in the Red Hat in the sector public. For me, is very important give this in meet, because Rolando and Efrain is a big partners in CNES and here in Mexico, and help us many initiatives of the technology and develop of not only the technologies and the solutions. If it's important, because the people, the learning and the knowledge is development too. And Efrain Sanchez is a solution specialist for Red Hat Mexico since 2011, and he works in Red Hat as consultant implementation with technologies of Red Hat and architect solutions. And finally, a specialist is very long experience with the work. These technologies make is to the nerve for all everybody, and not only on the Mexico, on the universities and public sector and research centers. Is very important your aportation in the communities of the Free Software and Free Software Foundation for Mexico. Ronaldo Cedillo has more than 25 years of experience on open source technologies, participating in multiple communities that include free software and Linux, kernel, Fedora, OpenStack, virtualization, containers, programming language, such as C, Python, Assembler, and etc. He worked on video gamers industry and IT developer and architect, specialized solutions, but hardware and enterprise software, high performance and environment, supercomputing, and distributed storage. He is a member of the foundation team of Red Hat and recently worked and Microsoft as director of technical audience, helping to evangelize the development of open technologies. In the agenda, Rolando talked about the operating systems in high performance computing or supercomputing. Let's go with Rolando.
Rolando, are you here?
I can't hear Rolando.
Let's see.
Ready.
Hello, can you hear me now?
We hear you. Thank you, Rolando.
You can hear me now, right?
Yeah.
Okay, great. Well, just one little question. And should I give a talk in English or in Spanish? Hello?
Could you repeat, please? I can't hear.
Should I give my talk in English or in Spanish?
In English, please.
Okay, yeah. Sorry, I wasn't really aware that I had to present in English, because I had like half of my presentation is in Spanish, so I apologize. I wasn't aware of this in advance, but no problem. I can still give my presentation in full English. No problem. I will translate the slides to English in real time for the people here who are not Spanish fluent, right? Okay, so allow me to share my screen now. I'm going to be sharing my screen. Right. Okay. Okay, you should be seeing my presentation now. Is that okay?
Yes, I see the presentation.
Perfect. Okay. So, well, thank you very much. My name is Ronaldo Cedillo. I'm a principal solution architect for Red Hat Mexico. And I was invited here to talk about Linux, to talk about the operating system, the role of the operating system, and the role of open source in supercomputing. And in particular, what's Red Hat been doing all these years to contribute in the open source, the free software, and Linux technologies in general, right? And so let's talk about what's the status and what's the situation here in supercomputing with regards to Linux. First of all, well, as most of you will know by now, Linux is just yesterday, I believe. Yeah, this is October the 5th, 1991.
There was the first version, the first functional release of Linux that was released almost to the day, 30 years ago. And Linux is this operating system that was created by this computer science student in the University of Helsinki. And that's very interesting because it started just as a very small project, but Linus Torvalds back then didn't know or didn't really expect to for this operating system to be basically dominate the world and technology in general. At some point, he jokingly said that this quote, I'd like to say that I knew this would happen, because that it's all part of the plan for world domination. Yeah, this was done in an interview when he was asked about if he knew or he wanted this to happen. He didn't at the time, but now in 2021, Linux is by far the dominant operating system in the industry, in the planet, just because most of the Android phones and a lot of IoT and most of the clouds run on hypervisors based on Linux as the operating system, and also, of course, the supercomputing landscape. If we go back 20 years, in 2001, Linux was not there in the top 500 supercomputers. It was not there in the field. It was, well, it already had like a very respectable, a sizable share of the landscape, with about 39 supercomputers out of the top 500 supercomputers in the world. And well, the dominant operating systems back then were all variants of Unix, right? And all the compute, like if we added or we basically all the sum of all the super computers, all the computing power together will be in the teraflops range, right? That was 20 years ago. So Linux was number 2, right? With less than 8% of the share of the top 500. But then just 10 years later, in 2011, Linux dominated by far the supercomputing landscape. Instead of just 8%, now it was 90+%, 91.4% of the supercomputing landscape. And well, by then, in just 10 years, the supercomputing power, the combined supercomputer power of all these top 500 supercomputers was about almost 100 petaflops, right? So we were in the range of petaflops by then. That was 10 years ago. And well, there were some other operating systems like Unix variants still were there. Some other open source technologies, and just one non-open source or non-Unix or Linux based or variation was there, like a Windows supercomputer, right? That was 10 years ago. But then what happened in the last 10 years? In the last 10 years, Linux completely dominated the supercomputing landscape. So we now have 500 out of the top 500%, top 500 supercomputers out there running Linux, right? And now we are in the range of exaflops, right? We again basically grew our supercomputer power in a new order of magnitude. Now we are in the thousands of petaflops and thousands of or hundreds of thousands or thousands or times what we had in 2011, just 10 years ago, right? And this is, of course, thanks to the advent of multi-core CPUs, thanks to the proliferation of GPUs, tensor units, a lot of different new types of hardware that are getting in the landscape now. And of course, all of those new things, all of those new technologies require basically an operating system, require a framework, require a standardized set of APIs or even ABIs that can run these tools, or can run this hardware, this pieces of hardware, right? So this is very important. Linux, this is no accident, of course. It's a very it's very important to say that it's a testament of how open source and free software helps the supercomputing community, because otherwise we would be still using proprietary and probably by now slower technologies that wouldn't be helping us to grow in terms of capacity, in terms of computing power, right? So this is definitely a success story, and it will be at least in the foreseeable future. Linux will keep being the dominant operating system, right? That's what I believe, and what most people in the supercomputing community believe also. And well, this is, of course, what we need to be able to grow from the exaflops range to the next level, right? Like zettaflops or jotaflops or whatever comes in the future, right? But then right now we're heading towards, I would say, some obstacles or hurdles in terms of the capability to grow. There are some challenges for growth. And in these challenges, among these challenges, I would say that this is a very important set of figures. Right now, we have two metrics that are very important in supercomputing, which are the Rmax and Rpeak metrics. Those represent the total of computing power, the actual computing power that is measured. That's the Rmax metric. If we go back to this slide, we see that the Rmax is about 2.7 exaflops, which is the actual computing power, the actual combined computer power of these two top 500 supercomputers. And then there's this other metric, which is the Rpeak. And this metric is related to the theoretical performance, the theoretical computing power that we could squeeze out of these top 500 supercomputers, right? But there's a gap in these two figures, in these two numbers. There's a gap. And the problem with that gap is that it's been growing steadily in the last 10 years. If we go back to 2001, the gap between these two metrics was 32%. What that means is that basically there's a 32% of optimization gap. There are many reasons for this, of course, and not least of which are applications and algorithmic optimizations, like optimizations that could be made at or could be done at the application layer, at the end user, or at the application layer. Of course, all the technological and technical hurdles to correctly parallelize any task or any job that we want to run in a supercomputer. This is very, very hard, of course, right? But the thing is, in almost 10 years, or probably more than 10 years, from 2001 to 2011, 2012, this number didn't grow. We were about 32, 33%. This gap remained basically the same in these 10 years. But in the last 10 years, from 2011 to today, which is a very recent update from the top500.org website, we have this gap, which is almost 40%, right? We grew from 32% to 39% in these 10 years, and this is very, very difficult to manage or to compensate, right? Why? What are the reasons why we have this problem? First of all, we have problems and challenges with the scheduling of jobs. There are so many solutions out there, so many ways to schedule jobs in a cluster or a supercomputer. And it's very difficult to choose the right tool for the job in many ways, right? And there's also a problem with the orchestration of services. Now, with the advent of self-service in the cloud, for example, and the combination of private and public clouds, which we now refer to as hybrid computing or hybrid cloud computing, it's become a problem just to orchestrate the instances, VMs or containers, whatever. We are also adding new layers of abstraction between the hardware and the application. Now we have containers. Containers is a tendency that is growing very fast. Is a tool and it's a technology that people are using increasingly. Virtualization, of course, we have more than 15 years in using virtualizations in supercomputers. And cloud computing, of course, are huge providers of supercomputing today, right? We can just rent or we can create instances on demand in the cloud, which are very cost effective. And they could be provided with GPUs and CPUs and tensor units, and we can scale in and out the infrastructure that we need to run our jobs, right? But this is very hard also, because we need to be able to provide the tools for people who are in the supercomputing community to be able to control these new layers of abstraction and to be able to translate or to map their jobs to this new reality, right? So this is very, very hard. Also, we have huge changes in the hardware architecture. A lot of changes in the past in the last 20 years. I would say just the last 10 years have been very critical in this area. Why? Because back in the 90s, we used to have supercomputers that used this memory architecture that's called NUMA, the non-uniform memory architecture. And what that had the ability to distribute jobs in huge scalable supercomputers. But that was very hard to do, of course. That was very difficult to manage, right? And that was kind of like the problem, the basic problem that we had back then. But now we are returning to that in almost every single type of computer. Right now we have NUMA on a single chip, like the current and future CPUs, and also GPUs and tensor units and so on, are being distributed as chiplet architecture. That means putting more than one chip on a die or on a PCB, right, on a motherboard. And that means that the memory in those computers is already partitioned by default. So it's very difficult to create jobs that can really be optimized for this partitioning of memory, right? This was not a problem 10 years ago, but it is today, and it's going to grow, right? And then there's also the problem of distributed storage, right? How to manage this huge, enormous databases and data sets, right? It's very, very difficult. So distributed storage is also a consideration that we have to understand in the reality of data centers, in the reality of the hybrid cloud, in the reality of virtualization containers, and so on, right? And self-service orchestration and provisioning, of course. So these are just some numbers. The processing lag or latency is increasing. That this is one of the problems with these new architectures and new models of provisioning hardware for supercomputing tasks. And there's a huge gap between each different how to say the connection between data and computing power, right? We can see that just a local cache inside a CPU or a GPU can provide maybe just a very fast memory, which is between 1 and 4 times slower than the CPU or GPU itself. That's measured in nanoseconds. Then the memory RAM or distributed RAM or NUMA would be in the range of 10 to 1,000 times slower, measured in nanoseconds to microseconds. Then disks, just getting data from a disk could be 10,000 or 10 million times or more times slower. That's measured in milliseconds or even seconds, right? And then there's the network. The network provides a great challenge, a greater challenge, because network can be very, very slow, like 1 billion times or more slower than what the CPU can or the GPU can process locally, right? That means, of course, that we have to be able to provide supercomputing that is distributed, right? We have this scalability and distribution problem, right? We have a centralized computing power. We used to have those. This is what the top supercomputers have. But if we see the whole landscape, we have a lot of different workloads and a lot of different use cases where this is not the solution or the right solution for the job, right? We need to be able to integrate cloud computing, either hybrid, private, public cloud computing. We need to be able to use edge or IoT, Internet of Things, and distributed jobs to localized computing units, right? Okay. Okay, I'm getting a message. Sorry for the interruption. They say, okay, a friend's telling me that I should switch to Spanish. Is that right, friend? Should I switch to Spanish now?
Sí, si quieres puedes cambiar a español, no hay
problema. Puedo cambiar a español. Bueno, vamos a repetir un poquito de esta parte en español. Muchas gracias. Bueno, estamos hablando de la latencia de procesamiento. Les decía, la latencia de procesamiento es una comparativa muy importante, ¿no? El gap o la diferencia entre dónde están los datos y dónde se procesan puede ser muy grande, ¿no? Desde cachés que pueden ser de la misma velocidad del procesador hasta la red que pueden ser miles de millones de veces más lentos en términos de latencia. La escalabilidad y la distribución del cómputo, ¿no? De tener cosas centralizadas a lo que hoy está en la nube, ¿no? Y también distribuciones de procesamiento a nivel de Edge, a nivel de IoT, ¿no? Y el ejemplo más claro de esto es el computer vision, ¿no? Las capacidades, por ejemplo, de reconocimiento facial, de inventario, logística y automóviles con autonomía, ¿no? Automóviles sin conductor. Esos son los escenarios más claros para un IoT, para un Edge, para un supercómputo distribuido, porque necesitan tener el cómputo localizado en el lugar donde ocurren las cosas y es cómputo que al final va a sumar dentro de un dataset o va a sumar dentro de una supercomputadora o un equipo, un trabajo que tiene esta manera de distribución y al final tiene un lago de datos o al final tiene un dataset que va a ser unificado, ¿no? Entonces es muy importante que entendamos que la realidad del supercómputo dentro del data center, exclusivamente dentro del data center, no es el futuro. Tenemos para muchos escenarios el cómputo distribuido que tiene que contemplar esta realidad, ¿no? La realidad de distribuir el cómputo en la red. Ahora, ¿cómo hacemos nosotros esto? ¿Cuál es la situación de Red Hat en esto? Red Hat tiene muchos años en el supercómputo. En muchas partes, evidentemente Red Hat no solamente hace supercómputo, Red Hat es, digamos que el proveedor de open source más grande del mundo. Y aquí hay solamente algunos cifras de años de experiencia que tenemos en todos los puntos que son requeridos para la realidad de hoy, ¿no? Por ejemplo, 25 años de soportar múltiples arquitecturas con el mismo código. Esto es muy importante para el supercómputo, tener la posibilidad de migrar mis cargas de trabajo entre diferentes arquitecturas, de manera que esto pueda sumar. Y hoy día, pues, tenemos una historia de soportar desde x86, Power, Spark, Itanium, cuando existía, ARM, MIPS, cuando eso también era más popular, ¿no? Y demás arquitecturas, ¿no? Entonces, todas estas arquitecturas que han sido parte de la historia del supercómputo en los últimos 30 años han estado presentes en Red Hat, ¿no? Tenemos unos 14 años, 15 años más o menos de contribuir en el kernel de real time. Esto es para algunos escenarios de supercómputo también. Es muy importante. Unos 12 años más o menos de incluir la virtualización en el sistema operativo base. Es decir, hace 12 años, un poco más de 12 años, que no distribuimos un sistema operativo que no sepa por default virtualizar, ¿no? Más o menos unos 10 años, un poco más de 10 años, 10 y medio casi, de incluir tecnología de containers. Hoy día la tecnología de containers está muy en boga. Hoy día es una tendencia muy importante el uso de containers en cualquier escenario de cómputo, supercómputo o no. Y nosotros tenemos 10 años, más de 10 años distribuyendo oficialmente un sistema operativo con capacidades de containers, ¿no? Ya sea a partir de tecnologías como el XC, como Docker, como Podman, como Kubernetes, ¿no? Por ejemplo. Tenemos unos 9 años de contribuir y de soportar OpenStack. OpenStack es una tecnología para clouds privados e híbridos. Entonces es una capa de IaaS y bueno, también puede ser usado para PaaS, para orquestación. Y todo esto con la capacidad, por supuesto, de hacer el autoprovisionamiento, el self-service de instancias, tanto de máquinas virtuales como de aplicaciones en containers, de forma que podamos integrar eso con un orquestador que pueda hacer el job scheduling, ¿no? Que pueda hacer la canalización de trabajos de supercómputo. Ahora, 10 años también con este producto que llamamos OpenShift, que es nuestra plataforma de containers, y 7 años de contribuir y de soportar a Kubernetes o K8S, ¿no? Como se conoce, Kubernetes es hoy día una tecnología que de la que todo el mundo habla en términos de implementación de containers. No estamos llegando, empezamos desde el principio en estas tecnologías y somos líderes en estas tecnologías, definitivamente, ¿no? 10 años de soportar soluciones de storage distribuido. Vamos a hablar un poquito más tarde más adelante del storage distribuido, pero tenemos soporte y el uso de storage para supercómputo desde hace muchos años. Desde hace más de 10 años de soportar tecnologías que hoy día son corrientes, que son importantes y reconocidas, ¿no? Hablando de TPFS, hablando de HPFS, hablando de Gloucester, de Ceph Storage. Primero que van a notar es que no está listado aquí Lustre, que hay muchos usuarios de Lustre en la comunidad del supercómputo. Voy a hablar de eso un poquito más tarde. Pero bueno, también hay escenarios donde nosotros hemos coexistido o combinado Lustre con soluciones de Red Hat. Bueno, 7 años más o menos de distribuir y soportar microdistros. Esto es para la optimización de cargas de trabajo, para instancias y containers y cosas mucho más pequeñas, de forma que podamos correr las aplicaciones que requieren supercómputo de una forma distribuida con mejor densidad o con mayor densidad, ¿no? Entre más podamos llenar un servidor de múltiples instancias y más podamos hacer ese particionamiento ligero de los recursos, más podemos llegar a cerrar ese gap que existe hoy entre las cifras de Rmax y Rpeak en las supercomputadoras o en el supercómputo que requerimos para la industria. Esto, bueno, tenemos microdistros, tenemos desde hace muchos años uno que se llamaba el Atomic Host, hace un par de años tenemos el CoreOS también, y lo que llamamos el UBI, que es el Universal Base Image, dentro de Red Hat, dentro del sistema operativo Red Hat, ¿no? Es muy importante. Ahora, otros 6 años de contribuir y de soportar Ansible, que es una plataforma de automatización muy fuerte y que es una plataforma que también está siendo tendencia, ¿no? Una tendencia clara también, no solamente la parte de orquestación y la parte de instanciar trabajos dentro de una nube o dentro de un escenario de supercómputo, sino también la capacidad de control, la capacidad de automatización de tareas dentro de una supercomputadora o un escenario de supercómputo es súper crítico. Entonces, la plataforma de automatización es clave para el futuro. Tenemos unos 5 años más o menos en las gráficas que vimos al principio, unos 5 años de los 10 que tiene Linux dominando fuertemente el escenario de supercómputo. Tenemos unos 5 años de dominar como Red Hat la familia de distribuciones Linux que se usan en el supercómputo. ¿No? Básicamente son Red Hat Enterprise Linux o las variantes que de ella emanan, las que hoy día dominan el escenario de las 500 supercomputadoras más grandes del mundo, ¿no? Entonces estamos ahí, somos un jugador súper importante en el supercómputo mundial y listo.
Roland, quedan 5 minutos para que le des chance a platicar a Efraín, porfa.
Ah, claro que sí, muy bien. Bueno, y bueno, vamos a... sí, de hecho, tengo bastante más material, pero bueno, vamos a sintetizarlo. Tenemos también entonces estas diferentes tecnologías, lo que es High Performance Computing, como vertical, es algo que que atendemos como mercado, ¿no? Capacidades de hyperscale o de hiperescalabilidad, evidentemente el Edge, el IoT, ¿no? La nube pública y privada, storage distribuido, microservicios y automatización. Todas esas cosas son las que nosotros hoy ya comprendemos dentro de Red Hat, ¿no? Y bueno, hay múltiples tecnologías, lo que son máquinas virtuales dentro del sistema operativo, arquitecturas donde podemos combinar containers con máquinas virtuales para podernos traer cargas de trabajo, ya sean legacy, aplicaciones que hoy día no son cloud native o cloud aware, y tecnologías que ya lo son, y poder ayudar en esa transición en un mundo donde hoy podemos obtener cómputo bajo demanda de todos lados. Esto es súper importante. Soportamos pues todas las nubes, básicamente todos los proveedores de nube que hay afuera. El Edge es una de las estrategias más críticas para Red Hat. ¿no? El hecho de hacer las imágenes cada vez más pequeñas. En muchas cosas que nosotros estamos haciendo dentro de RHEL para crecer la instrumentación del sistema operativo, poder ver herramientas de performance y cuánta cosa. La seguridad es algo que no podemos dejar, sobre todo en estos ambientes distribuidos, la seguridad se vuelve crítica, es súper importante. Ya no podemos no contemplar la seguridad como lo hacíamos antes en los 90s, donde, bueno, pues la seguridad era perimetral física, básicamente, y en un firewall, en un data center, ¿no? Hoy día ya no tenemos esos lujos, hoy día tenemos que pensar en ambientes distribuidos, en Internet, donde existen muchos más jugadores y muchos más puntos de debilidad, ¿no? Que tenemos que fortificar. Tenemos herramientas de containers, las distribuciones, como lo mencioné, tenemos una cosa que se llama la UBI Micro, ¿no? Que es una micro distribución de Linux, que es la Universal Base Image, una base pequeñita para containers. Estas capas de abstracción, como lo mencioné también hace rato, todo eso está dentro del sistema operativo y buscamos que cada vez sean más delgadas con el objetivo precisamente de reducir ese gap, ¿no? Arquitecturas entre containers y cloud, ¿no? Entre IaaS, PaaS o más arriba incluso, donde podemos hacer aprovisionamiento de aplicaciones en diferentes tecnologías o diferentes data centers. También tecnologías de containers para Edge, administrados desde una consola central, por ejemplo, ¿no? Containers distribuidos para IoT y cosas así, donde podemos tener clusters en diferentes tamaños conviviendo y dentro de un mismo scope o de un mismo alcance en la administración. Y bueno, Ceph. Ceph es hoy día nuestro storage distribuido estrella, ¿no? Ceph es una tecnología súper importante. Hace rato mencioné Lustre, que es uno de los storage distribuidos que usan muchas supercomputadoras en el mundo, o bien también, por ejemplo, HPFS o GPFS. Pero Ceph es una tecnología que nació siendo distribuida y que es completamente open source. Y hoy día es uno de los pilares de crecimiento de la parte de supercómputo, sobre todo por la cantidad de datos que se necesitan almacenar y la manera en la que esto puede crecer y también ser distribuido, ¿no? Ceph tiene capacidades de distribución donde puede utilizar también múltiples tipos de hardware por cuestiones de costos y por cuestiones de velocidad, ¿no? Es muy flexible en ese sentido. Y bueno, por último, ya para terminar y darle paso a Efraín, todo lo que hacemos en Red Hat tiene soporte multianual y soporte de muchos años. Nuestro sistema operativo tiene por lo menos 10 años de soporte. RHEL 8, que es la tecnología que tenemos más actual en sistema operativo, empezó en 2019 y tiene por lo menos hasta 2029 de soporte oficial, ¿no? Más otros 3 años de soporte extendido que pueden agregarse, o sea, 13 años en total, y todo eso pues le da a todas las herramientas que corren sobre Red Hat una estabilidad y una confiabilidad y capacidad de reproducirlo, que bueno, no tiene igual en la industria, sobre todo hablando de supercómputo basado en ellos. Muy bien, tenemos muchos partners de hardware, estamos en todas las nubes y con eso terminamos mi parte de la presentación. Muchas gracias.
Muchas gracias.
Muchas gracias, Rolando.
Definitivamente muy interesante tu plática. Ahora hay que dar la oportunidad a Efraín para que nos platique un poco más de OpenShift como plataforma de inteligencia artificial y obviamente cómo es abierta sus posibilidades, porque yo creo que lo que comentas es la base, pero ahora yo creo que es importante conocer dónde se aplica, cómo sacarle ese provecho, y yo creo que está interesante lo que nos va a platicar Efraín, que es su especialidad. Adelante, Efraín.
¿Qué tal a todos? Buenas tardes. Gracias por el espacio. Bien, vamos a ver algo que comentó Rolando, que también ha comentado el maestro Juan Carlos, y que es muy importante, no solo porque pareciera moda el uso de contenedores, sino porque hoy en día se está volviendo una necesidad para dar agilidad, para dar flexibilidad y sobre todo portabilidad a las cargas de trabajo que estamos generando en nuestros centros de datos y que a veces por temas físicos no podemos expandirlos de manera muy rápida o tan rápida como quisiéramos o lo demanda el negocio, ¿no? ¿Qué es importante para esto? Vamos a platicar acerca de 3 casos muy rápido sobre cómo con la plataforma de contenedores nuestra, que es gestión y orquestación de los mismos, lograron aprovechar adoptando temas de inteligencia artificial, de machine learning, de deep learning, para poder solucionar problemas y sobre todo el poder desbordar cargas de trabajo fuera de sus centros de datos cuando estos se veían rebasados. ¿Sí? El otro punto importante que vamos a platicar también es por qué escogieron a nuestros productos para estas tareas. Muy bien. La primera parte es por qué contenedores, ¿no? Si bien el tema de cómputo, al final, la abstracción de la complejidad de administrar todos los recursos que están debajo, no se ven ya hoy limitadas por el sistema operativo, sino por la operación misma que nos va dando el trabajo o el día a día. Si bien podemos con muchas herramientas automatizar el despliegue en bare metal del sistema operativo, generar clústeres de manera automática, preparar sistemas de archivos distribuidos para poder atraer, consumir datos, ¿qué sucede cuando esto empieza a salirse más allá de nuestro centro de datos, cuando tenemos que agregar hardware nuevo, y este hardware no está tan alineado a lo que ya teníamos? En fin, toda esa parte tiene que ser de una manera mucho más amigable. Ahora, ¿qué es importante para este punto? Los retos que mayormente la industria exige es, primero, el tema del retorno de inversión que no lo ven tan claro, ¿no? Sucedía muchas veces y me pasó con algunos clientes directamente que me decían, oye, pues es que el desarrollador me pide 4 petas de espacio para hacer un data lake y hacer pruebas, y pues eso la verdad es que no da un rendimiento, ¿no? Entonces, inmediato al negocio. Y esto es un gap a veces entre el científico de datos y el negocio, ¿no? Porque en realidad sí lo da. Hay ya tecnología que nos permite tener ese retorno de inversión más claro, pero no estamos llegando todavía a eso, ¿no? Ese es uno de los retos. Otro de los retos es la falta de talento. Otro reto todavía también importante es cómo la operación está aislada, ¿no? Muchos de los datos que se generan están todavía en bases de datos o en modelos legados, y traernos toda esa información también es un reto complicado porque son dos entes diferentes. ¿Qué otra parte también es importante? La falta de datos legibles, ¿no? Porque ya están contenidos en un set de datos, en un conjunto de datos organizados de una manera diferente de cómo lo necesitamos, y que también debemos de prepararlos para empezar a hacer todo el análisis, y eso representa también una dificultad. Y la otra dificultad también importante que se ve es qué pasa cuando nuestra infraestructura es rebasada, ¿no? Si nosotros queremos seguir los modelos tradicionales para aplicación o para casos de uso de inteligencia artificial y machine learning sobre almacenamiento tradicional, en donde tenemos un appliance con storage carísimo y del cual dependemos solamente de ese fabricante para crecerlo, ya alcanzamos el límite desde el momento en que lo compramos, ¿no? ¿Por qué? Porque un lago de datos definitivamente va a requerir mucho más espacio que lo que nos pueda ofrecer una sola caja o un solo appliance. Las compañías también nos han compartido, cuando están adoptando esas estrategias, que la complejidad para una adopción o para el viaje completo del despliegue de inteligencia artificial, machine learning o deep learning, está en todas las etapas, ¿no? No hay una etapa en la que parezca simple. En todas, la constante va a ser el tema de la infraestructura, que es constantemente demandada. No hay un solo momento en que esto no aterrice en esta parte. Entonces, si estamos observando que la parte crítica, parte fuerte está en cómo administramos o en cómo portamos rápidamente todo lo que estamos haciendo de un lugar a otro, esto nos va a llevar a la utilización de contenedores. Si bien las máquinas virtuales parecieran ser una forma fácil también de contener las aplicaciones, quizás no con la misma velocidad que lo haga un contenedor, ¿sí? Y con todos los procesos de Continuous Integration y Continuous Delivery, para todo lo que estemos haciendo con las implementaciones, supone otra complejidad de no solamente trasladar un contenedor, de no solamente llevarse el hardware o la máquina virtual de un sitio a otro, de un silo a otro, sino llevarnos toda la composición, todo el proceso, todo el knowledge de una manera organizada, ¿sí? ¿Qué vamos a ganar cuando estamos hablando de contenedores aplicados a temas de inteligencia artificial, de machine learning? Agilidad, portabilidad, flexibilidad y escalabilidad. Nosotros, como compañía, lo que vamos a hacer, y mencionando a OpenShift como plataforma, vamos a generar esa abstracción de la complejidad, no solamente de la gestión del hardware, sino también de la cuestión de procesos y desbordamiento de cargas. Teniendo la posibilidad de orquestar desde un solo punto diferentes silos o diferentes nubes privadas, públicas, hacer una composición, una gestión muy orquestada desde un solo punto de los diferentes silos que tenemos, sin necesidad de estar pensando o dedicándonos como científicos de datos o como los ingenieros de datos en tener esas herramientas, sino solamente dedicarnos a lo que realmente le tiene valor para el negocio. ¿Sí? Para ejemplos, tenemos ya en las verticales, en los segmentos de healthcare, de servicios financieros, de telcos, y como lo comentaba también Rolando, en los temas de la industria automotriz, en los temas de seguros, en donde no solamente han adoptado nuestra tecnología, ¿no?, sino han crecido los proyectos, han sido muy exitosos. Conceptualmente la arquitectura la vamos a ver de esta manera. La capa de lo que yo les he estado hablando en este momento es la infraestructura, sea en físico, en virtual, en nube privada, en nube pública o en edge. Lo que vamos a buscar siempre con el portafolio completo de Red Hat es facilitarles cualquiera de estos estadios, facilitarles o flexibilizar el poder llevar de un lugar a otro o escalar las cargas de trabajo, ¿sí? Como casos de uso ya muy puntual, tenemos el tema de salud. Básicamente, el problema era una respuesta rápida a la sepsis, que es una respuesta abrumadora a la infección que puede invadir a un cuerpo. Y la ausencia de detección temprana, pues evidentemente complicaba las cosas. Entonces, con el uso de nuestra tecnología, el desarrollo de todos los algoritmos para hacer el uso de inteligencia artificial, 5 horas, como lo dicen ahí, pueden salvar vidas, ¿no? Entonces es importante. La solución básicamente fue utilizar y analizar los datos de manera predictiva para establecer un enfoque estandarizado de detección temprana de sepsis. Los resultados que obtuvieron básicamente es un servicio de los más grandes que provee servicios de salud en Estados Unidos, es aprovechar toda la parte de inteligencia artificial para hacer un análisis de millones de datos y tener respuestas muy ágiles, ¿sí? Y todo en manos de los doctores, ¿no? Entonces, es un caso de uso muy importante porque las personas se dedicaron únicamente a construir todo lo que tiene que estar relacionado a su quehacer médico y no al estar tratando de ver, oye, ¿cómo ponemos o cuántos nodos de cómputo tenemos? O ya se nos acabó el storage. Ellos se dedicaron únicamente a la parte importante, a lo que era la ciencia de datos, y toda la parte de orquestar el hardware, de desbordar cargas, no lo delegaron. Entonces, esa parte creo que es muy, muy importante para todos los que estamos en este punto. ¿Qué otra cosa también en la industria automotriz? Todo lo que está haciendo esta compañía está desplazando o vendiendo cada año alrededor de 2.5 millones de automóviles. Y al tener esto, los datos que más le importan al final, al estar compitiendo con compañías nuevas como Tesla, como Uber, como Live, es básicamente tener conectividad con el usuario y brindarle una experiencia completamente digital, que puede ser desde el búscame un restaurante, ayúdame con la reserva, es generar toda esa experiencia del conductor al momento de usar un automóvil. Toda su plataforma, todo está sustentado con nuestra plataforma de contenedores, haciendo uso de inteligencia artificial, de machine learning, de deep learning, y se están olvidando realmente de qué hardware, en qué nube. La portabilidad para ellos es muy importante. Por último, el tema de ExxonMobil, que también están haciendo o utilizando inteligencia artificial para el análisis de datos y mejorar todos los temas de producción y optimización de sus operaciones. Por último, ya casi para terminar y respondiendo a las preguntas de por qué Red Hat, como pueden ver en esta lámina muy rápidamente, y ya lo comentó Rolando, desde el sistema operativo, desde la orquestación de contenedores, desde la parte de llevar la orquestación de hardware, ya sea por el método de infraestructura como servicio, por el método tradicional, hasta el almacenamiento definido por software, que es self-storage, y que viene muy bien con todas estas tareas que nosotros tenemos que realizar con la ciencia de datos. La razón de por qué Red Hat, una vez que ustedes se dedican, o que nosotros los que estamos en el desarrollo de algoritmos, de modelos, nos dedicamos a lo que nos importa y delegamos un poco el tema de quién o qué debe de estar orquestando el hardware, la capa física, básicamente es delegárselo a una compañía como nosotros, que seguimos siendo una compañía abierta, de código abierto, que cumplimos nuestra promesa de mantener toda nuestra tecnología de esta manera, ¿sí? Es decir, estamos siempre abogando porque las cosas que nosotros usemos, que estén en manos del usuario y de compañías de misión crítica, sigan siendo open source, ¿sale? Que la otra parte importante es el partnership que se puede tener con varias compañías de terceros de hardware, de software, y no preocuparnos por toda esa parte de cuándo vence, hay driver, no hay driver, funciona, no funciona. Es dedicarnos a esa parte importante, ¿sí? La otra parte importante es que cuando nosotros estamos utilizando una tecnología o una plataforma de contenedores, que esta plataforma, además de estar en pleno compliance con la Open Container Initiative, también puedan adoptar de una manera muchos lenguajes de programación que sea políglota, muchos proyectos o frameworks que ya nos permiten rápidamente interactuar con los modelos y no estar pensando en cómo integramos este componente, ahora si lo subo funcionará o qué va a romper. Y parte del trabajo de nosotros es el desarrollar arquitecturas de referencia en el cual nosotros facilitemos toda la arquitectura, toda la gestión tanto de los procesos como del hardware, para que la gente se dedique únicamente a lo que es importante para el negocio. Nosotros dejarnos en la parte de administrar y orquestar los recursos, que puede ser desde un data lake con Red Hat Ceph Storage, de un grid de memoria con la parte de InfiniSpan o Data Grid, el de poder juntar bases de datos relacionales, eventos, mediante Kafka o Is Hpc y no estar pensando en la complejidad de si se integran o no se integran, sino tener una arquitectura ya muy completa en la cual podamos estar desbordando cargas de trabajo, escalando rápidamente, sin complicaciones. Por último, el ecosistema es muy importante, el tener el respaldo también de otros fabricantes, el tener el respaldo de otras compañías que están haciendo inversiones muy importantes, que han tenido avances grandísimos en todo el tema de inteligencia artificial, aprendizaje automatizado, que no sea únicamente un proyecto que, además de ser comunitario, tenga un ecosistema limitado porque no lo esté adoptando o una comunidad científica o una comunidad empresarial, sino que tenga durante el tiempo de vida pues una longevidad importante. Y sobre todo que se permita la colaboración. Si bien todo el tema de las tecnologías abiertas se vuelve en el laboratorio de innovación, si estamos hablando de un tema ya de producción, de un tema en donde tenemos que resolver los problemas de una sociedad y que están demandando un nivel de servicio, pues entonces deleguemos esa parte de la integración, de la certificación, del ciclo de vida a una compañía como nosotros y dediquémonos específicamente a lo que importa en la ciencia de datos, ¿ok? Y para eso, pues bueno, nuestro portafolio es muy completo, que va desde la gestión del hardware hasta la integración del edge computing o de los dispositivos IoT.
Vamos a tener que cortarle, Efraín, para que le demos oportunidad a la comunidad de preguntar.
Por favor.
Una disculpa. Efraín, muchas gracias. Está muy interesante esto lo que tú nos dices y creo que pues da para más tiempo y una disculpa que los interrumpa, pero creo que es bien importante también escuchar la comunidad. Sé que también el quórum tiene muchas dudas, hay muchos comentarios, pero hay oportunidad de solventar algunas por el trabajo, la siguiente presentación que se va a dar así a continuación. ¿Alguna pregunta que tenga alguien? Me comenta que si nos pueden contar sobre la integración con Singularity.
No tengo ahorita en la mente la respuesta. Rolando, ¿tú has visto algo acerca de esto?
Sí, ¿sí me escuchan?
Sí.
Sí hay una colaboración importante con Singularity. Singularity soporta sistemas operativos basados en Red Hat desde hace años, desde hace tal vez unos, ¿qué serán? 7 años, más o menos, 7, 8 años, algo así. Entonces, sí es algo que funciona, vamos, sabemos bien que funciona, pero una de las cosas que creo que es el futuro, que ya lo mencionamos en la presentación, es que todo va hacia containers y hacia esquemas de cloud native. Entonces, aquí es donde viene el reto. Creo que ahí es donde está en este momento la transición con cosas como Singularity, pero es algo que sí va a pasar rápidamente, ¿no? Entiendo que ya hay un soporte actual para containers en Singularity sobre Red 8 y eso automáticamente lo pone en el mapa dentro de OpenShift, OpenStack y todas las tecnologías que ya tenemos actualmente basadas en Red Hat, ¿vale? Entonces, sí hay una, la respuesta corta, pues, es sí hay una integración, sí hay soporte y, digamos, tiene unos 2 años más o menos.
¿Alguna otra pregunta?
¿Una otra pregunta? Ok, bueno, si no hay más preguntas, yo quisiera agradecer como siempre la participación de Fred, que siempre ha sido en ese ámbito de colaboración y dar a conocer muchas de las iniciativas, no nada más tecnológicas para las soluciones de la industria y principalmente de gobierno, sino también en ese sentido de aprendizaje que pues nuestra comunidad académica en el desarrollo de las tecnologías aquí en México ha ayudado bastante. Entonces yo quiero agradecer mucho, como siempre, a Efren, a Rolando, como parte del equipo de Red Hat, y ojalá siga habiendo esta cercanía, este trabajo en equipo que pues, como se darán cuenta, pues hace falta mucho el expertise humano y bueno, agradeciéndoles de antemano a todos su asistencia y atención. Pasamos la palabra al moderador para la siguiente ponencia. Muchas gracias, Efraín. Gracias, Ronaldo.
Excellent presentation on Red Hat. As always, we thank you for your attention. Remember that you can also follow Carla 2021 on YouTube. And next, we now carry forward with a video by our sponsor, Altair. We hope you enjoy this video.
Hi, I'm the CTO for the PBS Works division at Altair, and I'm going to talk to you about the future of high-performance computing. I believe high-performance computing improves people's lives. It's why I love HPC. It's what wakes me up in the morning. It's why I've been in HPC for over 25 years. What does that mean? With high-performance computing, we can build more sustainable products, make the environment better. We can make cars safer. We can predict the weather to get people out of the way. We can use high-performance computing to do virtual tests of pharmaceuticals, something that's really important in today's world. It truly, truly impacts everything that happens in today's world. But what is HPC? Well, I define high-performance computing as doing more than you can do on your workstation or on your phone. It's taking computing to another level. It's not a particular technology. It's not a particular hardware. It's the idea of solving massive problems at a massive scale quickly. So where does it come from? Well, let me start way at the beginning. Okay, maybe not the very beginning. I'm going to start more or less around when I started. Here's a beautiful picture of a control data 6600. This is what the state of the art in high performance computing and supercomputing was about the year I was born. We won't tell you. You'll have to look that up. I don't want to say how old I am. But what characterizes high performance computing is that this machine was very unique, one of the first of its kind. It was pushing the envelope of how much computing you could get done. By the way, this computer today probably has about as much computing power as this earbud that keeps falling out of my ear. Maybe less. Maybe the earbud actually has more. I don't know. Possibly a parking meter. We're talking about a parking meter. So where are we going? What do we mean when we're saying high performance computing today? Well, we've gone from here to really absolutely massive scales of computing. The Oregon National Lab is building, slated for delivery next year, an exascale machine. What does that mean? Well, an exascale machine is capable of computing a billion floating point operations every second. It's an amazing accomplishment. It's a massive scale in order to do more science, in order to do better engineering, in order to really solve problems that are unsolvable today in a time frame that matters. And that's the important thing about high performance computing. Okay, so let me tell you about the future. If the past is about big computing and it's about these massive machines that only a few people can buy, what's going to happen in high performance computing? Three things are going to happen. We're building out, as we build out high performance computing, we're building it out in a way that more and more people in more and more ways can actually leverage high performance computing, can get access to it, can use it for more things. We're changing the way that you actually deliver high performance computing. And the high performance computing engine, the engine behind high performance computing is becoming way more diverse. Let me talk about each of those three things. There was a time when high performance computing supercomputing required PhD level knowledge in everything. You needed to have gone to school for many, many years in order to get any benefit out of it at all. But over time, and as we move into the future, we're building out things that one that used to be called high performance computing, but today you can do some of those things even on your phone in your pocket. And that's just tremendous. But one of the things that high performance computing future is doing is we're taking high performance computing and we're trying to make it accessible to more domains. Instead of just weather modeling, which requires these massive machines that run for days in order to predict the weather or to tell us where, by the way, I'm outside in front of my house here, to tell us whether the smoke is going to waft into this area or I'm going to have clean air to breathe. We're also taking high performance computing to other places, to individual, say, researchers or medical practitioners inside of hospitals, where they can immediately sequence DNA and build really fine-grained, personalized medicine. We're also taking it out. We're giving it to architects to build better cities, basically. So not just it's really expanding the scope. There's this big word that everybody uses called democratization. But what does it mean? It means more people, not just high-end super scientists, being able to leverage high-performance computing to really make decisions faster, design better products, and really get better solutions. So that's one. Two, the future of high-performance computing is completely changing in how we deliver it. It used to be that there was these massive machines that existed in one or two or, okay, maybe a half dozen or a couple of dozen places around the world. Well, these machines are huge. They're super expensive. They require a lot of power. What if you wanted to actually have access to one of these machines? Well, in the same way that although I own a house, I also, when I go on vacation, I actually rent a hotel room. There's this thing called, I'm sure you've heard of it, cloud computing, where you can rent access to computing. You don't have to buy the whole computer all the time. You can just rent it when you need it, just like renting a hotel room. And high performance computing is, as it moves into the cloud, and it will move into the cloud, but not completely. I'll say more about that in a second. As it moves into the cloud, it actually also expands what we can do with it and who can access it and where you can access it, because you can now access it anywhere, anytime, just through a network connection. But will we be all cloud like some people predict? No, of course not. Otherwise, this would be a hotel behind me, not my house. Of course, it's going to be a combination of some local people where you buy your own cluster and you have it and you use it. And then when you need more power or you need something that you don't have, maybe you want to take a good vacation and you want a jacuzzi bath and you don't have one at home, you go rent a hotel room. Same thing with high performance computing. And three, the nature of how high performance computing is delivered in terms of just hardware and software and technology is becoming hugely diverse. For a long time, high performance computing meant you go buy something that looks really a lot like your laptop does. Well, okay, maybe your laptop did a few years ago. It had a chip on it that it came from Intel. It had a little bit of memory, and it ran one of a couple of different operating systems. Probably Linux if you were doing high performance computing, maybe Windows, and that was it. And then what you did to build a high performance computer was you put all these together and you just connected them together with a network. Well, that is not true anymore, and that is becoming more and more diverse. So we now actually have really large systems that are built out of NVIDIA graphics processors. So originally designed and conceived to just do high-end graphics, it turns out that graphics processors are fantastic vector engines in order to actually solve some of the hardest problems in computational fluid dynamics, in AI. And so they're being applied. So now we have CPUs and GPUs, but there's other vector engines. Even NEC is rolling out their own vector engine, but it's not just Intel CPUs anymore either. We have ARM CPUs, we have Power CPUs, we have, it's an explosion of heterogeneous hardware in order to deliver the promise of high performance computing. But in fact, that's not even it. And one of the future things that people are looking at, and actually this is even available on the cloud today. I'm not going to call it high performance computing because it's too new, but you can actually get, if you've heard of it, quantum computing. But what is quantum computing? Quantum computing is just another way of processing data. Will quantum computers take over for everything? No, nothing ever takes over for everything. But actually, quantum is very good at some things, we think, if we can actually get quantum computing to scale up, to solve larger problems. Right now, although it's 40 years old, quantum computing is only solving the tiniest problems, and we're getting an inkling of what that is. But it just goes to talk about the diversity of all the different hardwares. In addition to bringing the hardware together and having the hardware be different, the technology in high performance computing, excuse me, is also amazingly different. The technologies we're bringing together is now we're not just solving physics problems using physics, we're solving problems also using artificial intelligence. So the diversity of how high performance computing is delivered, the engine behind high performance computing, is becoming way, way more diverse. So the future of HPC is really about more people using HPC. It's about different ways of accessing HPC, getting a broader way of getting access to HPC. And finally, it's about the bloom of diversity inside of HPC and how you can actually achieve it. But what about really in the future? Is it gonna fundamentally change? I've sort of said it's changing a lot, but really, the idea of high performance computing is not going to change. The idea of high performance computing is solve the biggest, most important problems lightning fast with the biggest computers and the biggest technology, the best technology that you have. So scale it up and get it done. Thank you very much.
Thanks to our sponsor, Altair, for this video. We hope you enjoyed it. We invite you now to continue with our last event, a special event of the day by the University of Guadalajara. We are very grateful for your presence, and with this, I say goodbye to you, and I will see you tomorrow on Carla 2021. We return now to Isidoro to present this special event. Cheers.
Hello, good afternoon to everybody. Buenas tardes a todos. Estamos ya en el evento final del día, un largo día que creo que